Welcome student for today's lecture. Student, today we are going to see the next point of our the biotechnology topic, that is the biological tools. In last lecture we have seen about the instruments which are used for the recombinant DNA technology. Today we are going to see for the recombinant DNA technology which biological tools are used. So, in the biological tools there are the three tools, that is first is the enzymes, then second is the cloning vectors, or it is also known as the vehicle DNA, and the third, that is the competent host, that is the cloning organism. So, the enzymes, then the cloning vectors and the competent host, these are the three main biological tools which are used for the recombinant DNA technology. So, first upon we can see that is about the enzymes which are used for the biological tools. So, which enzymes which are used, that is for the recombinant DNA technology. We see that is one by one enzymes. So, first enzyme is the lysozyme enzyme. See here first we see that is about the enzymes which are used for the recombinant DNA technology. So, for the recombinant DNA technology, enzymes, in which here the first enzyme which is used, it is the lysozyme enzyme. Lysozyme enzyme. So, this lysozyme enzyme, it mainly used to dissolve the cell wall, that is the bacterial cell wall. This lysozyme enzyme, it mainly used to dissolve the cell wall, that is the bacterial cell wall. Then the second that is used is the nuclease enzyme. Second enzyme which is used is the nuclease enzyme. So, this nuclease enzyme, it mainly used to cut the DNA molecule. So, in the nuclease enzyme there are the three enzymes, that is first, that is the exonuclease, then second is the endonuclease, and third is the restriction endonuclease. Third is the restriction endonuclease. So, see that is here the nuclease enzyme, it mainly used to cut the DNA molecule, in which here the exonuclease enzyme, it mainly used to cut DNA molecule from the 5' end or from the 3' end of the DNA, that is from only ends. So, see that is here the, if we see how the work of the exonuclease enzyme is, that is the exonuclease enzyme, here if we see the DNA structure, that is here there are the nitrogen base pairing, that is the DNA 5' end, 3' end, correspondence to this 3' end, here is the 5' end, correspondence to the 5' end, here is the 3' end. So, here if we see the work of the exonuclease enzyme, this exonuclease enzyme, it cut the DNA molecule that is only from these ends, that is from the 5' end or from the 3' end. So, when the DNA is cut, so here the phosphodiester bond is cut, and here the DNA, it is cut like this. So, it is the exonuclease enzyme. Then the endonuclease enzyme, this endonuclease enzyme, it cut the DNA molecule, that is, so see that is here 5' end, here is the 3' end. here correspondence to 5 prime end, here is the 3 prime end, and there are the nitrogen base pairing. So here endonuclease enzyme, this mainly cut the DNA molecule, that is from the middle portion, that is here the endonuclease cut, that is at any point except the ends. म्हणजे हे दोन एंड सोडून कुठल्याही मधल्या पॉईंटमधून जर DNA मॉलिक्यूल कट होत असेल, दॅट इज इट इज ड्यू टू द एंडोन्यूक्लियेज एन्झाईम. Here nuclease enzyme it mainly used to cut the DNA molecule. So exonuclease enzyme it cut the DNA molecule from the ends. That is, it may be from the 5 prime end or from the 3 prime end. Okay, so here it cut the DNA molecule, that is from the 5 prime end or from the 3 prime end. म्हणजे कोणत्याही एंड्स कडून हे एक्झोन्यूक्लियस एन्झाईम कट करेल. Endonuclease enzyme it cut the DNA molecule at any point except ends. म्हणजे एंड सोडून कोणत्याही पॉइंट मधून जर कट करत असेल, that is this enzyme is the endonuclease enzyme. Then third enzyme, that is the restriction endonuclease enzyme. It cut the DNA molecule in the specific sequence, that is in the palindrome sequence. In the palindrome sequence. So what is meant by the palindrome sequence or what is meant by the recognition site, we see detail see that is about the restriction endonuclease enzyme. तर रेस्ट्रिक्शन एंडोन्यूक्लियस एन्झाईम हे आपण पुढे डिटेल अजून पाहणार आहोत. तर आता सध्या फक्त मी तुम्हाला न्यूक्लियस एन्झाईममध्ये एक्झोन्यूक्लियस, एंडोन्यूक्लियस आणि रेस्ट्रिक्शन एंडोन्यूक्लियस हे 3 एन्झाईम्स असतात, हा कन्सेप्ट लक्षात घ्या. Then next enzyme, that is the DNAligase enzyme. Third enzyme is DNAligase enzyme. So this DNAligase enzyme it mainly used to join the DNA molecule. म्हणजे जेव्हा आपण DNA कट करतो ते परत DNA जॉईन करण्यासाठी, DNA मॉलिक्यूल जॉईन करण्यासाठी इथं लायगेज एन्झाईम यूज केलं जातं. दॅट इज दिस लायगेज एन्झाईम इट मेनली प्रोड्यूस द फॉस्फोडायस्टर बॉन्ड. सो हियर द DNA ligase enzyme it mainly used to join the DNA molecule, that is by the formation of phosphodiester bond. So this DNA ligase it is also known as the molecular glue. म्हणजे इथं DNA कट करण्याचं काम कोण करेल, न्यूक्लियस एन्झाईम करेल, आणि कट झालेला DNA पुन्हा जॉईन करण्याचं काम कोणतं एन्झाईम करेल, DNA ligase enzyme. Then next enzyme that is the DNA polymerase. DNA polymerase enzyme it is also known as the synthesizing enzyme. So here the DNA polymerase enzyme it mainly bring about the synthesis of complementary DNA on the DNA template. So DNA polymerase enzyme, this DNA polymerase enzyme it mainly used for the synthesis of DNA molecule. म्हणजे DNA टेम्पलेट वरती DNA टेम्पलेट न्यू स्ट्रँड फॉर्मेशन जे केलं जातं दॅट इज ड्यू टू द DNA पॉलिमरेज एन्झाईम. म्हणजे ओल्ड स्ट्रँड, म्हणजे जेव्हा आपण रेप्लिकेशन प्रोसेस पाहिली की रेप्लिकेशन प्रोसेसमध्ये 2 स्ट्रँड सेपरेट होतात, बरोबर आहे, आणि इथं 2 स्ट्रँड सेपरेट झाल्यानंतर नवीन स्ट्रँड जेव्हा तयार होतो तेव्हा DNA पॉलिमरेज एन्झाईम हा यूज केला जातो. या कन्सेप्टवरून लक्षात ठेवा, DNA पॉलिमरेज एन्झाईम इट इज द सिंथेसायझिंग एन्झाईम. इट मेनली यूज टू सिंथेसिस of the DNA, new DNA strand. DNA strand, that is the new strand of DNA. असं म्हणू आपण on the template DNA. And then the fifth enzyme, that is, it is the reverse transcriptase enzyme. Reverse transcriptase enzyme. So this reverse transcriptase enzyme, it mainly use the synthesis of DNA strand on the mRNA or the RNA. So here from mRNA, DNA synthesis takes place. That is the reverse transcriptase in presence of the enzyme, that is the reverse transcriptase. म्हणजे आपण ट्रान्स्क्रिप्शन प्रोसेस पाहिलेली आहे की DNA पासून RNA तयार होणं, mRNA तयार होणं, या प्रोसेसला आपण काय म्हणतो, ट्रान्स्क्रिप्शन. मग ट्रान्स्क्रिप्शनच्या विरुद्ध प्रोसेस इथं होते. म्हणजे काय होतं तर या mRNA पासून DNA सिंथेसिस होतं, दॅट इज नोन ॲज द रिव्हर्स ट्रान्स्क्रिप्शन प्रोसेस. या प्रोसेसला म्हटलं जातं रिव्हर्स ट्रान्स्क्रिप्शन प्रोसेस. आणि रिव्हर्स ट्रान्स्क्रिप्टेज एन्झाईम, की हे एन्झाईम काय करतं, इट मेनली सिंथेसाइज द DNA दॅट इज फ्रॉम द mRNA. म्हणजे mRNA पासून DNA सिंथेसिस होणं, या प्रोसेसला म्हटलं जातं reverse transcription process. And here enzyme that is used is the reverse transcriptase enzyme. It mainly used for the formation of or the synthesis of DNA from the mRNA. Then the next enzyme is the alkaline phosphatase enzyme. Next enzyme, alkaline phosphatase, sixth enzyme, alkaline phosphatase. So this alkaline phosphatase enzyme, it mainly used to cut the phosphate group which is at the 5' end of the double strand or the single stranded DNA. See, that is alkaline phosphatase enzyme. It mainly used to cut the DNA phosphate group. Cut phosphate group, which is mainly present at the 5' end of the double stranded DNA or the single stranded DNA. Phosphate group कट करण्यासाठी हे एन्झाईम यूज केलं जातं. So these are some enzymes which are mainly used as a biological tool. ओके, मग बायोलॉजिकल टूल मधला आपण पहिला कन्सेप्ट बघतोय, दॅट इज द एन्झाईम्स. ओके, या एन्झाईममध्ये आपण पहिला एन्झाईम बघितला लायसोझाईम. इट मेनली यूज टू डिझॉल्व द सेल वॉल. म्हणजे सेल वॉल डिझॉल्व करण्यासाठी हे लायसोझाईम एन्झाईम यूज केलं जातं. त्यानंतर न्यूक्लिएज एन्झाईम, इट यूज टू कट द DNA मॉलिक्यूल. याच्यामध्ये एक्झोन्युक्लिएज, एंडोन्युक्लिएज आणि रेस्ट्रिक्शन एंडोन्युक्लिएज हे 3 एन्झाईम्स येतील. त्यानंतर तिसरं एन्झाईम आहे DNAligase. इट यूज टू जॉईन द DNA, जॉईन DNA मॉलिक्यूल. म्हणजे कट केलेला DNA जॉईन करण्यासाठी, आणि जॉईन करताना हे काय तयार करतं, फॉस्फोडायस्टर बॉन्ड तयार करतं. त्यानंतर नेक्स्ट दॅट इज DNA पॉलिमरेज एन्झाईम. म्हणजे इट इज नोन ॲज द सिंथेसायझिंग एन्झाईम, अँड दिस मेनली यूज टू सिंथेसाइज द न्यू स्ट्रँड ऑन द टेम्पलेट स्ट्रँड ऑफ द DNA. म्हणजे DNA मॉलिक्यूल जेव्हा सेपरेट होतात, 2 DNAचे स्ट्रँड सेपरेट होतात, तर त्याच्यावरती नवीन स्ट्रँड तयार करण्यासाठी हे जनरली DNA पॉलिमरेज एन्झाईम यूज केलं जातं. रिव्हर्स ट्रान्स्क्रिप्टेज एन्झाईम, इट यूज टू सिंथेसिस ऑफ द DNA फ्रॉम mRNA. म्हणजे ट्रान्स्क्रिप्शनच्या विरुद्ध प्रोसेस इथं घडेल. आणि अल्कलाइन फॉस्फेटेज, इट मेनली यूज टू कट द फॉस्फेट ग्रुप विच इज ॲट द 5' एंड. म्हणजे 5' एंड ऑफ DNA और RNA यातला फॉस्फेट ग्रुप हा कट करण्यासाठी जनरली अल्कलाइन फॉस्फेटेज एन्झाईम हे काय केलं जातं, यूज केलं जातं. So these are some enzymes which are mainly used, that is for the recombinant DNA technology.
The construction of recombinant DNA molecules is a very sophisticated scientific technique involving the use of several key tools, one of which is restriction enzymes. These enzymes are isolated from bacteria that recognize specific sequences in DNA. Then, they are used to cut the DNA to produce restriction fragments. Restriction enzymes are assumed to provide a defense mechanism against several deadly viruses such as bacteriophages. They belong to a higher class of enzymes called nucleases, which have the ability to cleave the phosphodiester bonds between the nucleotide subunits of nucleic acids. The nucleases are categorized as exonucleases and endonucleases. Exonucleases remove nucleotides from the end of the polynucleotide chain. They hydrolyze phosphodiester bonds of terminal nucleotides, while endonucleases cut the DNA at specific points and are thus known as molecular scissors. Let's study how restriction endonuclease was discovered. In 1963, scientists Stuart Lynn and Werner Aber were successful in isolating the enzymes responsible for restricting the growth of bacteriophage in E. coli. One of these enzymes methylated the host DNA, while the other cut the foreign DNA. The second enzyme was called restriction endonuclease. Thereafter, in 1968, scientists H. O. Smith, K. W. Wilcox, and T. J. Kelly, while working on Haemophilus influenzae, isolated an enzyme called Hind2. Hind2 was the first restriction endonuclease whose functioning depended on a specific DNA nucleotide sequence. It was observed that Hind2 always cut DNA molecules at a particular point by recognizing the position of the 6 base pairs. Therefore, the specific base sequence is also known as the recognition sequence for Hind2. Today, more than 900 restriction enzymes have been isolated from around 230 strains of bacteria, each of which recognizes different recognition sequences. Restriction enzymes are named using a specific convention. Let's take the example of restriction enzyme EcoR1, which is derived from the bacteria Escherichia coli. The first letter of the name, E, is derived from the genus, while the next 2 letters, CO, come from the species of the prokaryotic cell from which these genes were isolated. R refers to the strain, while the Roman number indicates the order in which the enzyme was isolated from the strain of bacteria. Restriction endonuclease plays a crucial role in the construction of the recombinant DNA molecule. It recognizes a specific recognition sequence, so it binds to the DNA and cuts the double-stranded DNA helix at specific points on its sugar-phosphate backbones. The specific recognition sequence actually identifies a palindrome in the DNA. Observe that the following sequence reads the same on the 2 DNA strands in 5' to 3' direction and vice versa. In a palindrome, the base sequence of the second half in the DNA strand represents the mirror image of the base sequence in the first half. Due to this, in the DNA double helix, the complementary strand also represents the same mirror image. Restriction endonucleases cut the DNA strand slightly away from the center of the palindrome sites. However, this cut is between the same two bases on the opposite DNA strands. As a result, single-stranded portions are left at the ends. Moreover, there are overhanging stretches called sticky ends on each strand. Such ends are called sticky or cohesive ends because base pairing between them can stick the DNA molecule together again. Thus, restriction endonucleases play a crucial role in the construction of recombinant DNA molecules which have DNA from different sources. When the same restriction enzyme cuts different DNA molecules, all of them are left with similar sticky ends, which can be linked using DNAligase. Therefore, restriction enzymes are